Okay, let's start. Okay, evolution is composed of basically two basic components. If you make exactly same copy for our offsprings, once we have the same problem, then it's a very little chance we can survive. First component is diversity generation. Second component is natural selection. Right? These two are the main components. And diversity generation has a different method. Point mutation is one way. The other big invention for the diversity generation is sexual recombination. This is a part of diversity generation. Now, survival of a strong one. Now, the definition of a strong is ambiguous. In the evolution, it's not the strong one can survive. The survived one is strong. Means the survival is the ultimate virtue, and there's nothing about strong, good, perfect, complex kind of things. Okay. That's the point I wanted to point out here. Okay, very good. Identical twin and me may have 100% in principle. In principle, 100% genetic identity. Non-identical twin is the same as siblings, right? A sibling, there is a chance that maybe we have the same mother or we have the same father, but not sure, then there is some other possibilities. So non-identical twin means we were in the same mother's belly, then most likely I have the same father and same mother. Right? Then we can say this is the 50%. Even though my brother and me or my sister and me has a two node. One node goes to the father or mother side, and come is each one. Right? Two node. But still, it's not 50%, it's not 25%, it's 50% because we have a double connection, one to mother, one to father. That's why those combined effect, same parents effect, make the connection more closer. Okay, if we have only one side connection, then that rule of each one perfectly works. Then the relationship should be this, right? One node, half. Two nodes, half and half, half over half. Three nodes, 삼촌, uncle is 1/8 kind of relations. Queen bee and work bee, they're siblings. Both of them are female. Maybe they had one father and one mother, and they may have same situation of common siblings. And I gave the second question, how about the between work bees? I realized I saw some of your answers. And you didn't read the book, Selfish Gene. This answer I want you to hear. None of you gave that answer. So let's check what's going on. Female of B, they have DNA as a double copy, correct? 2N. We have a 2N information, correct? 1N is from father, 1N is from mother. We have two sets, one pair of chromosome, correct? Female of B is the same. They have a 2N, diploid. If it's a two set, we call diploid. That means we have a 2N of DNA. That means one pair of DNAs. If we generate sperm or egg, we generate only N, right? So this one is a monoploid, but they call haploid. Comparing to this is a half. So this is the N state, haploid. This is a 2N state. Full body like us, we have a one set, 2N. Very interestingly, male B has 1N. Still, they can grow up, and forming up to the full shape, and they are functional. And when the queen bee make the marriage flight, when they started to make new colony, and then the male bees follow, and maybe the last moment is going higher and higher. The winner, only one male bee, succeed to mate with the queen bee. Then queen bee collect those sperms from male bee and store them forever, maybe for 30 years. Then she use that sperm to make a queen bee, the work bees. Then, so there is a possibilities. Maybe the mating can happen with a multiple male. It's also possible. But some species, they mate only with the strongest one male. Then father is the same. And father has an N, then N can make the sperm exactly same sperm, right? So that means if they have the same father, their identities from father side, they have exactly same material. 50% is secured. And then the other half is from queen bee. It is recombinated and generate the different kinds of the eggs. That's a half. That's a 25%. So work bee and work bee, their genetic closeness is 75%, not 50%. That example is given in that book, Cypress Gene. I hope to hear some of you give that answer. 아무도 안 읽었어? 아니면 제대로 안 읽었구나? I hoped that answer, Jenny. I hope you guys read that book. That is a really interesting book. So the genetic, the closeness is very interesting in bees. Work bees, they are closer. It's in between identical twin and regular siblings. And they help each other. And they are closer than their mother. So then, if they are in trouble, who they may try to save, whether their queen bee or their siblings work bee. It makes more sense in terms of genetic, the identity. Maybe saving sibling may be better. The problem is none of them is fertile. They are sterile. They cannot make offsprings. But they have a chance epigenetic modification to change their work bee into queen bee. That's where we're going to talk next week. Anyway, the genetic, the similarity wise between work bee is 75%. That's an interesting observation. I have the 50% identity with my siblings. then if I save two of my brothers, then even though I sacrifice myself by saving them, then genetic amount-wise, it's the same amount remained, correct? That's kind of if we accept the idea that gene is a unit of survival and evolution, then even though I die, if I can save two of my siblings, it's equal. If I can save three, then I should die and save three. Similar situations, I have a daughter and sons. If I can save three of them, then it's 1.5 times than my gene. Then I can maybe happily can die by saving them. Cousins, they have 1/8 of identity with me. Then if I can save 8 of them, maybe that's equivalent, then I die or 8 of them die. By these calculations, maybe some observations why the people or animals or the lives, they shows us looks like unselfish the behavior. And even though they sacrifice themselves, somehow they help to survive or to save the same genetic informations. So that's kind of the logic of this selfish gene concept. Too easy? Still some of you said the T cell. So we have lymphocyte as a B cell and T cell. Antibody is made by B cells. About the T cells, we are going to talk later after midterm exam. Okay, very good. Wow, look at that. One antibody has approximately 150,000 the Dalton size of the shape. Those details we talk later. Okay, these two we are going to compare later. Germline antibody is not produced by naive B cell. Naive B cell cannot make antibody at all. If the B cell make antibody, that means they are exposed to antigen and they are activated and they becomes plasma cell. It's a matured B cell, and then they start to make the antibody utilizing the original information we got from mother and father. So that antibody is what we call germline antibody. Once they make the germline antibody, they start to make another diversity. There's so-called mutation, a very high speed of mutation, very high level of mutation. That's why this is called as hypermutation. Okay, and then this property we discussed later. Okay, let's move on. Today, before we start the class, I invited Hongju to introduce the small concept of cDNA library. So this is the basis of the concept we are going to talk next class. So Hongju. Okay, please welcome Hongju.
Hello, my name is Shin Hongju, and today I'm gonna talk about cDNA library. So, in our cell there are lots of DNA sequences, but not all the sequences are transcribed at the same time. But only few of the sequences are transcribed at the same time under a certain condition. So in same cell, there would have different transcription. at one given point in time, which means they can have different cDNA libraries in the same cell. Likewise, different types of cells such as neurons, skin cells, stem cells, and so on would have different transcriptions too, because different genes are expressed in each cell. So, simply speaking, cDNA library is like a pool of mRNA at a given point in time under a certain condition. So, what is the difference between genomic DNA library and cDNA library? First, genomic DNA library contains all the sequences, both exons and introns. But cDNA is synthesized from mRNA, so it only contains exons, the protein coding genes, no introns. So a genomic DNA library is representation for all genes, while cDNA library is like a small subset of the genomic DNA library. Then how can we prepare cDNA library? First, we can get RNA molecule from target cell or tissue. After RNA isolation, we can get cDNA by reverse transcription. Let me give more detail for it. So this is mRNA molecule which is processed. Processed means introns are removed and exons are ligased, which means matured. And this mRNA is modified. For example, we can have poly-A tails like this, AAA, at the 3' end. Then we add oligo-DT primer and reverse transcriptase. Then we can synthesize cDNA molecule from this mRNA. So this cDNA is complementary to this mRNA. Then we remove mRNA by using alkali such as NaOH, and then we add DNA polymerase to synthesize the second strand of the cDNA. So after that, we use an enzyme to cut this loop, and finally we can get double-stranded cDNA. So back to this content, we can amplify the cDNAs by PCR, and we can insert the cDNA into vector, this bacterial plasmid, and this recombinant DNA is transformed into bacterial cell, and we can grow the bacterial cell in certain media. And after that, we can observe some colonies here, and each colony has same DNA molecules. So we can finally get cDNA from these bacteria. So how can you use this cDNA library? First, functional screening. It is identifying genes that are involved in specific functions. For example, we can get cDNA library from cancer cells, and then we can introduce those cDNAs into normal cells, and cells that express certain phenotype can be selected. So in this way, we can identify the cDNAs that is responsible for a specific phenotype. Another use is studying epigenetics. Actually, I don't know well about epigenetics. That will be covered in next class. But one of the famous concepts in epigenetics is methylation. So we can get cDNA libraries from cells with methylation and without methylation. So we can compare those two libraries and then finally identify the difference between the two cells in terms of expression. This is the end of presentation. Thank you for listening.
Any questions for Hongju?
Okay.
Okay.
Before the slide, it shows the S1 nucleus purple loop.
Yes.
I don't know the exact mechanism how purple loop. If that has a problem of the loop, and how can S1 nucleus change the loop? Like if it has a lot of bulge or like structure has a mixed together. So
enzyme would break down the bond and yes, help the make double strand. And 나머지 퀘스천은 제가 이해를 잘 못했어요.
이불 자를 때 루프가 되게 미용적으로 크게 늘어나고 아니면 꼬렸을 때 이제 S1 불면서 다른 소리를 표현하는
잘 아는 부분이 없는데 혹시 이거에 대해 아시는 분이 계시다면 대신 설명해 주실 수 있나요?
Okay, she needs help. Anybody can answer that question?
What's the question?
What is the question?
Sorry, I'm not good.
Okay, here, seems like those, in the beginning there was a mRNA. Okay, in mRNA case, 5' is the starting point, C' is the ending point of the sequence. So that's why we call at the end of mRNA, usually they have multiple A's, poly A's, okay, out of A, B, C, G. So that's why we call them like a poly A tail. So that means that this is the head and this is the tail. So if there is a template using reverse transcriptase, now utilizing the RNA as a template, they can make teeth with the DNA going this way, and then maybe primer you can use a poly T. They can bind to A's. So you can use poly T as a primer and synthesize this DNA strand. DNA, RNA, which is more stable? DNA is more stable, and RNA you can destroy by enzyme or chemicals. And DNA is saved, and then RNA is removed, chemically cleaved. And then remove them, and then if you utilize this as a template and make the other copy, then you need to have another primer this side, right? So originally it had a poly A tail, it can be used as a primer side, like primer sequence the position, but this side can be random, so it may be difficult to recognize it here. So there are two choices. Utilizing maybe there's a partial overlapping like this, and native partial the hybrid, or you can use random primer. So by using random primer, and then you can make anyway, starting any positions and make those copies. So two ways. So assuming that there is a small loop like this, and then if the synthesis goes go wrong, then the other side is perfectly packed. This is more rigid, so this is more protected area, and this is maybe a little open like this. There's a more susceptible to be cleaved by non-specific integrations. So that is kind of an example of how to cleave this long one into two strands like this. There are several options to overcome that in the synthesis. Okay, other question? So let me give you a question. This cDNA library is genomic library, or transcriptomic library, or proteomic library? It should be transcriptomic library, because the genomic library is covering all the DNAs, which may include intron and garbage DNA and everything. They may have its meaning, but this one is only representing mature mRNA, means which can make the protein cells. So in principle, this information is for proteomic, but proteomic has different modifications, post-translational modification. So this information is matching to mRNA. Collection of all mRNA, and which is different between different cells. So one cell type specific mRNA collection is matching to cDNA. Okay. So this is some basis the material we are before we go to epigenetics. Okay, other question? Okay, if not, thank you all too. And as you see, I invite any of you guys volunteering for giving small talks, 5 to 10 minutes talks. You can look up to those topics from this week. Next week, epigenetics. The next week, iPS stem cell. And after midterm exam, there is antibiotic, anti-cancer, and different topics. You can volunteer. You can send me email or the Kakao Talk. And then I wanted to give a small talk like this. Introduce something, interesting and not boring, not too much technical, kind of a small talk like this. Okay? Okay, let's just start. I will go to my slide. Okay, this week we are going to study about the genetics and evolution. So first of all, what is genetics? Well, genetics, it can be taught genetics 1, genetics 2, maybe a whole year topic. Evolution, maybe 1 semester. Molecular evolution, another semester. So this is about the 2 years of the class we finish in 1 week. We can do it, right? So let's briefly check the contents of genetics. In genetics, what do we handle? First of all, genetic information, how it can be transferred to the next generation, and how those informations are expressed. Expressed means how it is transcribed and how it is translated. So what kind of information is stored where we already studied. Genetic information is stored in DNA. And when they are expressed, we utilize RNA as temporary information system. And then eventually the end product is a protein. And how they are transferred to the next springs, next generations. If the life form does not invented sexual system, means half and half of 2n to 1n system, the only option is by making exactly same copy. And then transfer. Maybe it's more efficient. That's why bacteria can divide 3 times an hour. We can divide maybe most active cells, like cancer cells in our body, they can divide only once a day. That's why, in terms of number generation, we cannot compete with the bacteria. But there is another advantage we can utilize, how we can survive better in the competition, and the kind of a topic we study in generation to generation in evolution. So we, in a sense, seems like when we make our software springs, we make our copies, but actually it's not exactly same one. We are keep changing all the time. And by adapting into the environmental change. And when we talk about evolution, as I told in the quiz problem, usually we have the concept that maybe from simple to complex, from immature to mature one, or from weak to strong one, or from different component into more complex form, not always. Genetic evolutions, the direction is only defined by whether we have a better chance to survive in the given environment. And which direction we are going to move to, we don't know. Currently, maybe Homo sapiens is maybe most successful the species on this the planet, but which direction we may go, we do not know. That depends on what kind of change we encounter. And sometimes those complexity, it's better to drop. We sacrifice some function and save that energy and use that for the other way. Maybe that's better for survival. So again, not the strong one can survive. The survived one, as the result, we call it is strong in terms of survival. Okay. Now during the evolution of the process, the basic 2 components I said, diversity generation and selection. And for the diversity generation, for bacteria, it just makes its a copy of DNA. And only way it can generate the diversity is mutation. And those mutations is naturally also occurring. But if you have very harsh conditions like UV light or hot temperature or food shortage kind of shock from outside, then they tend to generate more errors when they make their copies of the DNA. So that kind of pressure can accelerate those mutations, especially in very difficult the environment. They generate more mutations than peaceful time. But when we observe this recombination power, if you generate this complicated system of sex generation, now we have a lot faster way to make diversity, and even one generations we can generate, one person can generate either sperm or egg, and 23 times of divided the chromosome has a chance to generate this number of different kind of sperms or different kind of eggs in terms of genetic information. So that means if you have only one chromosome, all the genes together in one cell, then maybe the option is only two, right? A copy or B copy. Because this is distributed into different chromosomes, and then they can be randomly shuffled, and then any one of those options can go into one sperm or one egg. It can generate about 8 million combinations. So 8 million different kinds of sperms can be generated. Now, that sperm meet eggs. Eggs also has a similar number of diversity like this. Even though we have the same father and same mother. Now, my brother, my sibling, and myself, maybe I made some technical mistake in the question. My brother and I have exactly the probability of exactly the same genetic information. If I'm female, then chance is 0, right? So I have XX, my brother has XY, there's no chance to be identical. Assuming that we are same XY, then still the combination wise 8 million by 8 million, 64 trillion is the chance. 1 over 64 trillion is the probability of whether we have the genetic information. Of course, the general structure of A and B and A prime and B prime 98% identical, but those 2% of difference make those diversities among us. Especially human beings, if you look at those animals, usually those siblings look much more closer to each other than humans. Interestingly, Asian and Caucasian, we have about 2% difference in genes. But Asian and Asian, Korean and Korean, also we have about 2%. That means human being has much more individual diversity than even different other races. Different races, of course, we have some common part. That's why we look a little more different. But among same races, also there are a lot of diversity in humans. So this power of a combination, so combinatorial power, make the diversity generation is even more than exponential. You cannot compare this power of a recombination to the point mutations. Okay? So this is the resources of generating diversity. It's not all. Even in the same chromosome, when we have the meiosis, means when we make the sperm cell or egg cells, they divide into two first. This is parent, this is one pair from father, one pair from mother, and they can change some part of that chromosome too. So it generates more diversity. This kind of a technique is what the observation, what we call homologous recombination. Another recombination. Homologous means this one and this one is almost identical. That's why they attract to each other. And then somehow they can change from the same position. They make more combination than 23 pairs of chromosomes. Okay? By these powers, it's quite difficult to find my date partner and have a chance to have a mate and make offsprings. That others can be rewarded by this power of diversity generations. That's why those sex are the mechanism that containing the life forms has much better chance to survive in a changing environment in the history of evolution. Okay? Okay, so that is kind of the individual level or the species level, usually what we call evolution. Now, if you look at our immune system, there are kind of a micro scale or cellular level, evolution is always ongoing. So this is the example of that system. Antibody is generated by B cell, and antibody, when this B cell, before it meets antigen, then B cell is not activated. Only when it meets some strange material from outside, most likely. In the beginning, when B cell is made and circulated in our body, and they do not interact with our own part. If they do, then it makes a problem, so-called autoimmune disease. They should not make antibody against our cells. So once it meets non-self foreign body, then that foreign body we call antigen, and then this B cell is activated, and then mature B cell we call plasma cell, and it starts to make antibodies. Now, one plasma cell, it generates only one type of antibody. Okay, one plasma cell make one antibody. That means this is like one individual of us. And different plasma cells, they make a different antigen, different antibody. Now so let's look at the antibody first. So antibody shape is like this Y shape, and seems like it has altogether 150 kilodalton, one heavy chain here, another heavy chain here, identical, and then one light chain here, one light chain here. Now, this protein is called as immunoglobulin. And then molecular weight wise, the 150 kilodalton, and it seems like the size wise, this one looks quite similar, right? So this is approximately 50. This is about 50. And one heavy chain from here to here, this is also about 50. Then one light chain, can you guess? 25 approximately, right? So it's easy to memorize those size. Heavy chain 50, light chain 25. Functionally speaking, here, this area, this is antigen recognition part, and this is constant part. So this part is called as FC. In the video I said that that's not F constant. What does that see? Crystallizable. Crystallizable because it's a constant. So that's why easy to make a step, right? So this area is AB, antigen binding area. So this part is called as FAB. This is called as FC. Okay. So then 50, 50, 50. And antigen binding site here. This part is antibody side. This is called as paratope. And the other part, antigen side of binding to antibody, what do you call? It is called as epitope. Okay. So, okay, let's move on later. Okay, this is one question you gave in the general questions. What is the role of a constant region of antibody? Can they bind to other protein? If you look at this structure, the connection is through, this is one strand of protein, another one. Four of them are connected together, and those connection is made by these two disulfide bonds, one here, one here. Role of this, now you can guess. Without this, they can be disassembled, go apart. So this constant part, it can hold two of them together. And natural antibody, this part and this part is identical because they are made from same genes. Okay, so this constant part can assemble these two, and also it can help the forming this structure. And with this FC, the binding affinity of FAB is further increased. It's just stabilizing this part. That's the role of FC. And second part, this is interesting point. Can they bind to other protein? it can bind if you inject this antibody. Let's say this is a human antibody. If you inject this human antibody to mouse, what do you think happens in mouse body? Mouse will recognize this as antigen, isn't it? Because it's a human protein. Then mouse will generate antibody against human antibody. And then this human antibody is a huge protein. That's why mouse antibody can bind to here, many positions. That means one human antibody has many epitopes. And mouse antibody can generate multiple antibody which can bind to different site with a different affinity. Right? Some of them may bind to Fc area. Then it's possible that if you generate this antibody as tool, then this antibody may bind to, let's say, virus. Now, if you use that mouse antibody, which recognizes this one, and then that secondary mouse antibody is labeled with a fluorescence motif, then it can be used as detector for this antibody. So that kind of a molecular tool can be generated. So the point is, one antibody for one species, our antibody can act as antigen to other animals. Then they can generate antibody to our antibody. And that antibody, mouse antibody, if you inject that into rat, then rat will generate third antibody which can recognize mouse antibody. Okay? So that kind of thing can be used as a research tool for this. Okay, another question for monoclonal antibody. Anybody can answer this question? Our body can generate many antibodies. Even though we take only one protein, we can try to bind to many positions, generate many proteins. Among them, we wanted to get one antibody which may bind to that antigen most strongly. Then that single antibody we call monoclonal antibody. And to make the monoclonal antibody, there is some technique like this. So spleen cell means here, how can the fusion of a spleen cell and myeloma cell occur? Myeloma is kind of a B cell like cancer cell. Cancer cells usually they live for long or forever. And here it says spleen. How can spleen cells have a semi-permanent characteristics? Is that the genome of myeloma cell move to the spleen cell? Well, there's some the mixed concept here. That spleen cell means there is B cells. Spleen has B cells and T cells. 70% B cells and 30% T cells. So here it said spleen cells, but that is primary regular B cells. And regular B cell and tumor B cell is fused. Those fusion is membrane to membrane. You need to give some shock, maybe electric shock, with high vibrating electric shock. Maybe some of those membranes are fused. And then they share information for antigen, means antibody making information is in this B cell, and the cancer cell, it provides the ability to live forever. So two cells fused together. This is kind of artificial chimera cell. And then this B cell, usually they cannot survive for long, but now they obtained the ability to live forever and to make antibody. I said one plasma cell is one individual which can make one type of antibody. If you have a chance to isolate that cell and then multiply them, and it's exactly the same genetic information, right? Those kind of cell, what do you call? Clone, okay? Because they have the same genetic information. Here, our interest is the genetic information to make one antibody. That's the genetic information. Second question. How does that selection can be successful or unsuccessful? For the biological process, if you want to select only one character of cells, here you have cancer cell and you have primary cells and primary B cells. Primary B cell cannot live too long. Cancer cell, they can live forever. If you mix them, only 1% of cells have formed those hybridoma, let's say, hybrid cells. Then the cancer cells, maybe the population-wise is even bigger, they may occupy most of those space, right? Then that's difficult to get the cell what you want. Then you can give some tricks here, one cell and two cells. Maybe you can give, one way is you can give antibiotic for both of them. They can die. Only when they combine together and they have two components together, then they can survive. If you design your experiment that way by giving two antibiotics, and only when they are fused, they can survive against that double antibiotic. If it's a single each of them, one antibiotic is okay, but the other one can kill leftover all others. So let's say you have 100 B cell, 100 myeloma, mix them together, only 1% fused. 99% are original cells. Now you give two antibiotics. The other 99% of cells all die out. And only this 1%, they can survive and come back. So that's kind of a common the trick. So this is the one way to answer this question.
About the first one you just said, the original T cells that are not hybrids, they cannot live very long. So can we just? I don't think we need two antibiotics, but only the ones that kill not hybrid, not fused myelomas, then we not fuse cells.
Technically, that's a good point. So I just tried to give you just a simple example. Assuming that one cell has a one character, the other has a different character. Only when two characters are combined together, and then they can survive. Even single ones, that they cannot survive. So that's kind of just the simplified version of the explanations. Technically, you need to think a little more the details about it. Okay, let's think about the genetic connections. Antibody is a protein, right? That means antibody should be made, or the antibody information should be recorded in DNA, right? Then we generate millions of antibody. Then how many genes do we have? Is the question. And when we learned about the Human Genome Project, we assumed we may have many genes, maybe million, or at least maybe 10% of million. But surprisingly, we have only 25,000 genes. It's too small to explain, our complex personality and culture and whatever. We thought we are much more complicated. But this molecular level here, antibody number is million. Then how many genes do we have to make that antibody? That doesn't explain. We have only 25,000 genes. And then the answer was, seems like this binding position, it has three components of different small building blocks. Heavy chain has 3 building blocks. Light chain has 2 building blocks. And it seems like those genes are recorded in the genome, one block each, 51 here, 27 here, 60 here. Interestingly, when we were born, the sperm cell has this whole gene, but when they are differentiated into B cells, among those, during the maturing process, they take only one each from here, and then make these combinations. And finalize the B cell, it carries only one cell of this. And same thing for the light chain, it gets only one cell of here. So, if the originally 51, 27, 6, then it can be multiplied. Heavy chain can make about 8,000 different heavy chain. Light chain is also about 200 types, and this can be combined together to make one antibody. Originally sperm cell, one cell, we know it, right? When they move to differentiate into the B cells, they are matured. Matured means they give up a lot of those majority. They save only one copy each randomly. It's similar to making sperm cell recombination, isn't it? Out of 23 chromosomes, take only one to make one set. Here, out of here, hundreds of different sets, take up only one each. And then if you multiply this together, amazingly, five-dimensional combination, it can make 1.6 million. This is half a set. Half a set means that this is maybe from father. We have another set from mother. Maybe you have 3 million. So this is what we call germline antibodies. This is the information we inherited from our parents. So this germline antibody is different between different person. So my germline set and your germline set is different. Maybe this number is also different. I have a 51 here, you may have a 50. So there is a small differences. That's why some people has stronger immune than other people. Anyway, in principle, in general, we have about millions of different combinations by this recombinating of different informations. Then among this process, then if you look at those gene number, how many genes here? 51 plus 27 and plus 6, 84 genes. Here, 40 plus 5, altogether about 100 genes, 100 different positional genes. This is what we have. Out of 25,000 total genes, maybe this is some combination of genes we devoted, assigned to make antibodies. Only hundreds, but if you use that as a Lego block, building blocks to make diversity, then it can make the millions. So that is the power of recombination, which is very similar to sexual recombination, but this is the happening in cellular level. And the end result, generating diversity. And then the next step is the selection, right? Selected one, now once we generate millions of them, now we get the coronavirus. And our body try to raise the antibodies, millions of them. If it is partially successful, then maybe those strong the B cells, they survive and all others are removed. And then those cells, now they try to make another diversity. First step of germline, the germline antibody, now they have these genes combined like that. Now for each of those positions has pressure to generate very fast mutation. That's what we call hypermutation. That's maturation step. So first stage, they select best binder. And then second step, they make more diversity very quickly. Multiple mutations every point. And then they get matured and further. And that end result is what we call somatic hypermutated antibodies. So molecular level versus cellular level of evolution is ongoing. And if you compare this into key and lock model of the enzyme binding to substrate, now we have two models, right? Key and lock is very rigid key, and lock, they bind like this. Induced fit, originally it's an open structure, but when it binds to the target, it changes the conformation and then wrap it up like this. That means induced fit is more flexible, right? Key and lock is more rigid, but it's more specific to the target. So when they studied those structure of antibody, they found that germline antibody is kind of open structure. So even though it has not fixed form, it can bind to many different shapes. It can bind to this one, it can bind to this one, it can bind to this one. So this is like a germline shape. When they recognize, now they try to make it more and more rigid, and then they don't move much. And during that evolution process, they move from induced fit model to key and lock model. Okay, so this is some of you, oops, correct answer, right? So germline antibody induced fit and is moving to somatic hypermutation and coming to key and lock and stronger binding and smaller diversity. It's more defined the directions. So these cells later they can survive for long as a memory cells. And this process takes about 2 weeks. When you get the invader and original the germline cells are get activated, general soldiers came out and then try to select which can fight better, and then hypermutation over the other week, and then they finally make very specific strong binder, and those viruses removed. Then they save very small numbers of a specialist as a memory cell in the body. Next time when we get the same virus, then these cells multiply very quickly by saving those 2 weeks and then respond much quicker. Okay, that is the cellular level of evolution we observe. Now, molecular evolution, even smaller size. Well, evolution is kind of combining process of mutation to selection. And mutation can happen to DNA level, and that is individual level. That is what we call general evolution. Species are changing over the time. And we just have seen those cellular level of evolution is also possible, like antibody generating T cell. T cell also has similar things, but we are going to talk about it later. And it can go down to single level of protein, even not the cells. Now, in cell case, this mutation and selection during the path, you need to make multiply those cells, those whole cell level of biological process, proliferation, making copy of the cells, make up those population, right? If it's in cellular level, if it's an antibody, that's also protein, but multiplication is done by cell level. But if it's protein level, just a single protein, then still you need to have genetic information, DNAs. That idea, if it's applied to molecular level, we can call it molecular evolution. One example is utilizing virus. We discussed a little about whether virus is alive or not, but the virus has a DNA inside, or RNA, let's say DNA, and then it has its protein product, and outside is a protein is coated. Then if you can introduce diversity to this protein, then we utilize those DNA information here and machinery system here. It's not cell, it's not life. Still we can use this as the information system and machinery system. And this can be amplified by the bacteria. And you generate the diversity here. DNA, you can make random DNA and you can insert it here. It can generate the different proteins, like antibodies. And then you amplify this utilizing bacteria. And then you break down those bacteria, then it generates many copies of a virus. And you select out the best binding to this and select them. And then that virus, you can reinfect 2 bacteria. So that's kind of a partial selection. And then the virus can be further amplified and generate maybe more diversity. And then further and further evolved. I asked you a question that what is the difference between individual evolution and this virus, the evolution, and any better option? Well, I didn't hear the answer I want you to hear. In our case, in B cell case, the to make antibody, when they make the right antibody, they can survive better. In our case, if we have a proper mutation or proper DNA, we may survive better. So that's kind of a cellular level or individual level of survival. But in this case, they can use only this binding event. They generate the protein, and then utilizing fishing pole, and then just take out the best binder. So this is a molecular level So it's difficult to do survival of a virus or not. It is just taking out by utilizing this binding affinity. So this is much faster, but this one is in a sense, this one is kind of limited only to binding-based screening. There is kind of limitations. If you go further to smaller size of molecular evolution, now all you need is DNA and protein. Even you don't need a virus, right? This kind of system is also possible you can design. So using DNA or using RNA and making protein, what machinery do you need? Virus case, virus has infection to the bacteria, and then it can kind of self-replicating, right? We utilize that one. Now, if you have a DNA here, you have protein here, then to amplify this DNA, what do you need? How can you amplify DNA? DNA polymerase, and what do you call that reaction? DNA replication is amplifying the DNA altogether. It's a PCR, right? If you have a machinery of combining those together, DNA polymerase with a template and building blocks, and if you can utilize a PCR, you can mimic cellular replication. And then if this can make a protein, then to make a protein, what do you need? What is the machinery to make a protein? Ribosome, right? Then you can add the ribosome here. Then ribosome can read this information and let it make protein. That is what they call ribosome library. Ribosomal library. That's the smallest molecular evolution unit. You have genetic information one side, and the other side machinery system means protein, and that is even ultimate small size of the molecular evolution. Okay, that much. I think this is end of the slide today. Any question for today's lecture? Today we have briefly reviewed about genetics and evolution. Genetics, how to store our genetic information that's in DNA, and expression into mRNA and protein. And evolution is between generation when they transferred to the next generation. Almost the same genetic information is transferred, but always there's a small changes. And those changes either by mutation or recombination and make a diversity and then transfer it. So based on this genetics implies that our characters and our traits are inherited from parents. But at the same time, we hear many times, our character can be affected by environment. So that environmental factor, how it affects to our genetic system is epigenetics. Okay, so that is the topic that we are going to cover the next week. Okay, so let's stop today's class here. If you have any questions, then you can come to me. Okay.